Good afternoon. Welcome back to Gen Bio 115. This is topic 9. We're doing cell respiration. So I want to give you some outcomes to start out with. We're going to talk about cell respiration, obviously, but I'm not concerned about you knowing every single number of every single molecule or drawing a bunch of molecules for me. What I'm really worried about is the general process. We're going to spend a lot of time looking at where the electrons go as I'm oxidizing and reducing. How do I phosphorylate ADP into ATP? But I'm not worried about every single enzyme that's involved. I want you to be able to make a flow chart sequencing what we talked about in this video. I want you to compare and contrast the 4 stages of cell respiration. And then I want you to break it. What happens if there's no oxygen? What happens if we can't make pyruvate? So ask yourself what happens if a piece is missing, what can still occur, what no longer occurs. And finally, I want you to make a big chart, and I'm going to ask you to do this in workshop as well. Make a big chart and ultimately compare similarities and differences between photosynthesis and respiration. Some things are very similar, some things are very different. The one thing I absolutely need you to do is no more than what everyone tells me they learned in high school. What is the mitochondria for? He's the powerhouse of the cell. Let's explain why he's the powerhouse of the cell. I think you know this, but now you're going to have to tell me why things happen. And that's the last time I'm going to say that. Our practice question from the end of photosynthesis: where do the electrons go? I will ask you this on the exam. I want you to be able to trace electrons ultimately from splitting water in photosynthesis all the way to making ATP in cell respiration. So I'm going to ask you to build me a really long sequence at the end of this. But for now, we're just getting them from NADPH, which is created in what process? Light or the Calvin cycle? The light reaction to G3P, which is the final product. It's the final acceptor of electrons in the Calvin cycle. So let's go ahead. I'm going to break this down into more, I think I have it into 5 different parts of the lecture. I'll do an introduction, and then I have a video for each one of the major stages of cell respiration. We've seen this formula a couple times. We've seen the formula for photosynthesis. Right now, I want you to look at your photosynthesis notes and write down on top of the photosynthesis formula, write down the formula right here for cell respiration. In this formula, we're taking the sugar that was made in photosynthesis, and in the presence of oxygen, we're going to do some stuff with some enzymes. We're going to have some metabolic processes, and the byproducts, the things that are given off, are going to be carbon dioxide. That's what you breathe out. There's some metabolic water. This is not enough to make you live, but it's water that's given off as a byproduct. And a lot of energy. This energy is going to be ATP. That's our adenosine triphosphate. We're going to build them as a process of breaking down sugar. This is a bunch of redox reactions that end in a way for us to phosphorylate. If you are having trouble figuring out redox reactions, sometimes it helps to follow the hydrogens. This molecule of sugar, C6H12O6, loses its hydrogen, it loses electrons, and it ends up as just CO2. So, what is oxidized here? Oxidation is losing electrons. Sugar is oxidized. When I learned this the first time, I just went around practicing sugar is oxidized in cell respiration. And that helped me remember the rest of the process. So if sugar, oh no, I ruined it. If sugar is oxidized, what's reduced? We're going to take oxygen and reduce it into water. Water comes out at the end. The electrons from sugar end up in the oxygen, attached to oxygen with their hydrogens. They're in the water at the end of this process. We're oxidizing sugar and reducing oxygen to water. Remember redox reactions? We have to pass the electron from something to something. You should always have a place where your electrons are going. Don't just throw them out into space. So in a eukaryote, and we're looking at eukaryotes specifically here, but that does not mean that prokaryotes cannot go through cell respiration. They absolutely can. But I'm going to do specifically eukaryotic respiration here because we're going to do a lot of, well, we have a bit of a eukaryotic human bias, so we do a lot of human stuff. Here, we're going to start out in the cytosol, in the cytoplasm, and then we're going to finish our process in a mitochondria. The goal is to oxidize a glucose, a single molecule of energy-holding sugar, the one that we made at the end of photosynthesis, and produce a lot of ATP by moving electrons and protons. The protons are hydrogen ions around. So we don't really need this whole picture. We're gonna zoom in on this part here, and we're gonna kind of look at two parts today in this part of the lecture. We're gonna consider how does the sugar get into the cell, and then how is the mitochondria actually involved? I have a little bit of questions I think you should answer before you go on, though. If you can't answer these, I want you to go back to metabolism, go back to membranes, go back to photosynthesis, and see if you can answer these questions. I didn't type the answers to all of them in here so that you can have sort of a blank list of questions. So sugar is oxidized and the electrons are passed to ultimately the final product, water. And we use the energy in those electrons to end up with a lot of ATP at the end. So what's the source of electrons for cell respiration? That's the glucose. We're going to break this down. These glucose electrons have a little bit of their bond energy. It has a higher Gibbs free energy than our final product. Is this an exergonic or endergonic reaction if I'm going to break sugar apart and release energy at the end? It's definitely exergonic. How about this molecule here? Is it polar or not polar? It's polar. It's definitely polar. Glucose dissolves in water. You try dissolving sugar in water. It mixes well with water. If it's polar and it's large, hypothesize how it gets through the membrane. What are the possibilities? So quick, go back to your transport lecture. There's a couple ways it can get through, but diffusion and osmosis are not ones of them. Simple diffusion. Oxygen is going to help drive this process. The oxygen that you're breathing is actually the thing driving the entire process once we start to do our steps in the mitochondria. So why is oxygen a good molecule to pull on electrons? I'm going to put it at the very end, and I'm going to have it pull electrons through the mitochondria. Why is oxygen good at that? Also, how does the oxygen get in? This is a small, non-polar molecule. How did it get in the mitochondria in the first place? And finally, where did the glucose come from? How did you get it in your cell in the first place? What process makes the glucose? And where does the oxygen come from in this process? What process builds lots of oxygen gas? We will answer those in review. I want you to get comfortable with those. You should be able to, I'm not going to write the answers here. I think these are things you should already know. And if not, this is something we should definitely discuss before you go on and really try to get into the meat of cell respiration. I guess you get in the sugar of cell respiration. You should also know about these electron carriers, these electron donors in this process. In previous lectures, I've introduced NAD positive plus some electrons plus a proton, this hydrogen ion. I build them together and I created NADH. This process of NAD positive plus 2 electrons plus a proton, I give electrons to NAD positive it accepts them. It's an electron acceptor. And I build this molecule. This molecule's job is to carry an electron from one place to the other. The electron's a little excited. It's still got a little bit of energy in it. And I need to take it from one side of a mitochondrium to another. There's a similar molecule right here, FAD. FAD is also an electron carrier or acceptor. When I build hydrogens and electrons, it becomes an electron carrier. FAD is only found in eukaryotes. You won't find it in prokaryotes. But it's a lower energy electron carrier than NADH, but still pretty good at carrying electrons around. They represent sort of a stored way of using energy in those electrons so that I can take it someplace and use that energy, the movement of the electron energy, to help build ATP, to synthesize a molecule of phosphates that I'm going to be able to use to drive a lot of different kinds of cellular processes. What was ATP? Right, I got my super simple drawing here. Again, I've been, even in practice sessions, I've just been making this a giant A. I don't care about you being able to draw it accurately. I care about you drawing these three phosphates. So I have this adenosine, and it's a triphosphate. Where's the energy here? It's there in that spring-loaded bond. These phosphates are all negatively charged. That means they want to get away from each other, like charges repel. And as they get away, this one is easier to pop off than you would expect. But it still has energy in this bond. This is an easily available source of energy for your cell to use, and your cell is good at it. How did we put energy in this spring-loaded bond? Like, how did we load it? I mean, that's cell respiration. That's the rest of this. So we've got all our molecules. We're going to add in some proteins. I'm going to definitely talk about some enzymes. You should be comfortable with the fact that enzymes are proteins that are reusable, that lower the activation energy. Check over your notes a little bit. See what we've talked about in the last 3 weeks. Just glance over it. And then so that everything's kind of fresh and swirling around, maybe start some long-term potentiation. We'll do a bit of everything. And then walk into part 2, 3, 4, and 5 of this lecture so that you're comfortable with the building blocks. And then we can synthesize them together to talk about cell respiration. And I'll see you in the next video. All right, bye-bye.